Hi, this is Mr. McLean, and in this session, I'm going to talk a little bit about how different chemical reactions within cells need to be coupled. So we're going to look at how an exergonic reaction can be coupled to make an endergonic reaction possible. So when we start this off, we see that cells have capability of producing or doing three different general categories of work, and we've already discussed some of these in previous chapters. Specifically, we talked about the sodium-potassium pump as a form of active transport. We've talked about mechanical usage, such as muscle contractions and the use of motor proteins in cilia and flagella. So the general idea of the energy coupling concept is that you're going to have something that is energetically favorable, and that's what an exergonic reaction is. So exergonic is going to have a negative delta G value, and endergonic is going to have a positive delta G value. And the idea is that if you use the energy that's released from an exergonic reaction, you can make an endergonic reaction possible. And the molecule that's most commonly used for that process is ATP. So we're going to take a look at how ATP can act as a shuttle of energy. So when we start off, first thing we want to do is we want to take a look at what the general structure of an ATP molecule involves. So you're going to have your adenine component over here. You're going to have the sugar ribose, which is a 5-carbon sugar, and then you're going to have these 3 phosphate groups that are going to be bonded together to make this the triphosphate. And what we're going to see is, in order for us to actually release some of that stored potential, chemical potential energy, we need to have a hydrolysis of the ATP. So we're literally going to take ATP, we're going to add water to that system, and by doing that, what we're going to do is we're going to split the third and second phosphate bond right there. So that's going to leave us with a molecule of ADP and a molecule of inorganic phosphate, and we're going to have a net release of energy. So this is an exergonic reaction. It's also, if you think about it, it's catabolic, because you have something that's more complex, and then as we go through that more complex structure breaks down into 2 smaller components with the phosphate and the ADP. So a lot of times when we look at different kinds of reactions, we mentioned that there was a phosphorylation that took place, and by adding that phosphate to a molecule or to a protein, we will see changes take place. Okay, so let's look at an example that we had talked about in class, and this is a scenario where we have something that is going to be coupled. So our initial reaction of concern is we want to form glutamine. All right, so in this example, you're going to take glutamic acid, chemically combine ammonia to it, and you want to form glutamine. So under experimental evidence collecting and data collecting, they've determined that the change in free energy is definitely going to be endergonic, and that's going to have a quantity of positive 3.4 kilocalories per mole. So that would indicate that this is not spontaneous. So this is not something that would be energetically favorable. It's going to be requiring an input of energy. So if we take a look at it, we often say, well, ATP is our energy source. So let's take a look at what ATP is actually going to do. So we're going to invest ATP, and the hydrolysis of the ATP is going to yield that inorganic phosphate and the ADP molecule. Now, when the phosphate attaches to the glutamic acid, what that does is it puts it in a condition of less stability. So when we have a less stable molecule, then it's kind of like what we had talked about before. When you have something that's at a higher point of instability, even if you have to do the activation energy, it will be favorable to progress towards an area of greater stability. So this phosphate ends up being displaced by the ammonia. So when the ammonia comes in there, it boots out the phosphate, and we have the molecule that we were looking for, which was glutamine. So if we take a look at it from an energy calculation standpoint, we see the first part of the reaction, which is what we figured was going to be a positive 3.4 kilocalories per mole energy needed. And we see this, which is the hydrolysis of ATP. Okay, and the hydrolysis of ATP under standard conditions has been calculated to give us a negative 7.3 kilocalories per mole. All right, that's a value that we told you should be familiar with. Now, also remember that when we're talking about a cellular environment, this value actually increases and goes to a negative 13 kilocalories per mole. All right, and that's because of the concentrations that can be found inside of a cellular environment that are different than what standard conditions have. All right, so bottom line, in order for this reaction to take place, we have to have a net negative energy change. And in this case, we do. We have a negative 3.9 kilocalories per mole, so therefore this reaction is possible. So this is a good example of something that's going on inside of our cellular environment that is a coupled reaction. So we're going to use ATP kind of as the connection point between these two different things. And as we said, when you hear the term phosphorylation, that means that you have transferred that phosphate to some other type of molecule or component in that situation. So when we think about ATP, we understand that ATP may be the energy molecule or the energy-rich molecule. I use the analogy of your cell phone batteries. When the battery is fully charged up, it would be like ATP. But as we do work and utilize that battery power, it releases energy and it breaks down. And eventually, when you have your battery full of ADP, that's the low energy state. It's not going to be able to do any work. We want to reinvest energy into that through other processes to reestablish ATP. So as we look at cellular respiration and photosynthesis, we're going to see the ADP ATP cycle quite a bit, and that's going to be an important idea. So looking at how ATP is so important and being able to couple reactions together really emphasizes the need to have efficiency. And when you take a look at living organisms, they rely on biological catalysts, which are enzymes, and that's where section 4 comes in at. So if we look at this example right here, we have the disaccharide sucrose, and this alone right here spontaneously can break down into the two monosaccharides that make that up. But it doesn't happen quickly because spontaneous doesn't mean fast. So it might sit there for almost an extended period of time beyond what we could possibly wait for. But with the utilization of the enzyme sucrase, we're able to do a hydrolysis reaction and break that down into two different parts. So when we think about any type of reaction, a couple things that we need to know. First thing is there's usually going to be some type of initial energy needed to start things up. We refer to that as our E sub A, or the free energy of activation, or more simply the activation energy. And a lot of times that's going to be a heat source. This graphic is pretty important. It shows you that we take our reactants, which in this case right here have more energy than our products. That's why we have that negative delta G value. But it tells us that in order to take these reactants to the transition state, we have to invest energy. So we push them up this mountain to the activation point where we're at our transition state, and then we allow them to break down into the products. So enzymes catalyze reactions by lowering that barrier. Okay? Enzymes make this pathway easier to achieve. So you would maybe see a pathway that has a much lower peak that would be needed. And things to remember, enzymes do not change delta G. So they don't make it higher or lower. They don't put energy into the system. They do not get consumed by the system. So when we look at these two pathways right here, the red path is going to show us that by utilizing an enzyme, we're able to actually carry out this reaction in a more efficient and reasonable time frame. So what we notice whenever we get into any of these different scenarios is we're gonna see a transition that's going to occur here. And the first thing that has to happen is we have to have what's referred to as an induced fit. So if you look at our substrate molecule right here, the substrate does have a matching active site, but it doesn't fit it like a lock and key. Rather, it squeezes in there and it contorts and it changes the active site. That's what the induced fit was. The other example or analogy would be to kind of clasp somebody's hands in a clasping handshake, to kind of contort that to make that fit together. So when we look at it, enzymatic reactions lower activation energy. And what they do is they do a couple things that are really critical. Number one is they get proper orientation. So in order to have these substrates undergo chemical reactions, we want to make sure that things are oriented in the appropriate position. So once it's inside that active site, that's one of the first accomplished objectives. Number two is the twisting and contorting will actually strain some of those bonds in that substrate. So when you strain the substrate bonds, it puts it in a position where you don't need as much invested activation energy to make that break. We're going to find that in some of these instances, the pH may be optimized in a lower range than what can be achieved in the cytoplasm. So by creating a microenvironment, what that does is that gives you favorable opportunities for this chemical reaction to take place. We also see the possibility of having covalent bonding to the substrate. So all of those things, and when you look at this this graphic right here, we're going to see that the first thing we do is we induce the fit. We have substrates that are held in there by weak different types of interactions. We have hydrogen bonds and we have the ionic bonds that are playing a role. The idea is that the reaction is increased, or the speed of the reaction is increased, because we lower the activation energy. So by lowering the activation energy, it makes it easier for us to take these substrates and convert them into the different products. Once the products are released, you're going to notice the enzyme is now restored back to its original configuration, which is ready for new substrates to go in there. So the enzyme does not get consumed or used up in the reaction in any way. Now, the environment itself can have a couple different impacts on that. One of the things that we're going to notice is that we have to worry about denaturing the enzymes. So inappropriate temperature or pH can definitely have an impact on the functionality of it. And beyond just the denaturing, we also find that they function better at different optimal ranges. So for example, we can look at different species and we can see that certain enzymes will function better at different temperatures. So in this case right here, we have a heat-loving bacteria, and it really functions at a temperature much, much higher than anything that humans would have. Humans peak out right around our optimal body temperature, which is around 37. So when you start to push higher, we see that it's not functioning, and we see the same thing when we push lower. Same thing about pH. Even within a human, we've got two examples right here. We have one that's optimized for the stomach, and we have another one that is optimized for an intestinal tract. Okay. Recall that there are a couple other things in here. We have the non-protein helpers. So review the cofactors and coenzymes as we finish that up. And I'll finish the last section with a discussion about inhibitors. Hope that helps.